for giving me this opportunity to present here in this AICTE Training and Learning Academic sponsored FDP. Uh, I am Kamlesh Kurankar, Director of KWebEx Info Solutions, and I am working in the field of biosciences and artificial intelligence. I will be taking you through this astounding journey of recent contribution of AI in biotechnology. We will explore the groundbreaking advancements made by AI, particularly with tools like AlphaFold, and how these innovations have transformed our understanding of protein structures. This presentation, which I am going to present here, will also discuss some ongoing challenges, ethical considerations, and future directions in this exciting intersection of technology and biology. I, will, I feel myself very fortunate that I have this mixed knowledge of biology as well as technology. So let's begin our journey. The title of my presentation is The Intersection of AI and Protein Sciences, a New Era of Innovation in Biotechnology. And I have specifically chosen a case study which is recently happened in the past few years that will give you an idea what are the possibilities and capabilities of AI in our biological field. Tejas, is my video visible?
No, sir, the video is not visible. I think you are sharing the presentation.
Okay, just a minute. Just a minute.
Yeah.
Let me share it. Is it now?
Yeah. Yeah, it is visible now.
Okay, okay, okay. So before proceeding further, I would like to start with this quote, famous quote: "When we will understand the physics and chemistry of the cell, the biology will be over." So that's what we will understand throughout our lecture. Actually, I was planning to take some poll. Yes, poll about what my audience are, whether my audience is related or having some knowledge of bioinformatics. Unfortunately, the poll didn't load here. So may I ask the audience just to raise your hands if you have the at least some knowledge of bioinformatics or AI, or are you using it? If you have, if you your answer is yes, you can just raise your hand because the chats is not available. Yes, I am seeing it. Snail Shinde ma'am. Okay. Anyone else? Okay, I'm seeing so many hands. Okay, ठीक है. Okay. You may take down your hands now. ठीक है, so that I can get the idea how deep I can go or how basic level if the knowledge is required, I can go through it. Okay. So what if I tell you there is a fundamental phenomenon which was existing in this universe since the billions of years, and recently it has got a plausible answer, and also scientists throughout the world who were bothered to solve that question, to solve that biological problem, they were trying for about 50 plus years, and it that problem has got solved in very recent years, and the problem was so phenomenal that several Nobel Prize were awarded throughout so many years to solve those problems. And this breakthrough is actually going to change the entire biological society, the biological sciences, the way we are looking at it, and the kind of application we are going to use or produce the outcome using that technology. So I guess that is the one thing fascinating for all of us, and that starts with this uh, phase. I have divided my presentation in several phases. I'm calling these as phase zero, the basics, where I'm just going through the basics quite fast because I'm sure all of you must be knowing these things. But still, just to take a quick review, we all know that proteins, which are very important biological molecules, they are so essential. Without them, we cannot imagine our life. They play a very critical role in virtually all biological processes. And those proteins, they are composed of amino acids. We know there are 20 different kinds of amino acids. And these amino acids, when joined together in a particular fashion, in a particular kind of sequence, they produce a functional three-dimensional structure that we call as a functional protein. So understanding these protein structures is vital for uncovering the mechanisms of diseases, designing new drugs, and advancing biotechnology. This slide sets the stage for our discussion on how that AI is transforming the field of protein science. These proteins, they have this hierarchical structure, which is shown here on the right hand side of the slide, starting from the very simple chain, that is primary structure, which folds into two kinds of different secondary structures. We know them as alpha helix and beta sheets. Then there comes a completely three-dimensional functional structure that we call it as tertiary structure. And sometimes more than one monomer interact with each other to form a completely multimeric protein. We refer it as quaternary structures. Proteins are so diverse. Proteins ranges from few amino acids up to several thousands of amino acids. For example, I have put here one TAL protein, which we are seeing here. Is transcription activator like protein, has only few amino acids, about 33 to 35. And one of the largest protein, which is titin, as the name itself suggests, has thousands of amino acids. So proteins are very diverse in their nature and very diverse in their function. Here, I'm just putting some functions that proteins carry out. Proteins act as an enzymes. We know the vital part of our metabolic systems. Proteins act as storage material. They say, for example, amino acids. Proteins are secreted as through the glands as hormones. Some proteins, they are in our muscles. We call them motor proteins. Then there are very important kind of proteins, that is defensive proteins, which are protecting our body against pathogens, for example, antibodies. And then there are these transport proteins which carries molecules across the cell membranes. Then there are again these receptor kinds of proteins, which we were very much familiar because of the recent outbreak of that pandemic. Okay. And then there are these structural proteins. No doubt there are other types of proteins also. I have just put some important kind of proteins here, just to show you that how diverse proteins are and how important they are for our life. So basically, if we see where the story begins, it actually begins during the early stages of this earth, when the prebiotic soup, the primordial soup, sometimes called, it was developing on the earth when the earth was gradually cooling. So those were the era around 3.7 to 4 billion years ago. The proteins were started evolving at that time. And since their emergence, the proteins have been constantly evolving, adapting to new environments and functions. And this evolution has been driven by natural selection, as we know that, the Darwin's law, and that results in the diverse array of proteins we see in all forms of life today. The proteins are also evolving as we are evolving side by side. Now, one thing uh, might be all biological students may be knowing that structure determines function. Very small sentence, but has a very important place in biology. The structure of the protein determines its function. Okay, here are some examples I have put. For example, this SARS-CoV-2 spike protein, the serotonin receptor protein. Here is the hemoglobin protein, which helps in the transfer of the oxygen across the body. The cytokine kind of receptor, the collagen, the luciferase protein, and there are other proteins. Okay. So with that basic knowledge, let's come to our phase one, the problem. The problem that scientists, people, researchers, biologists were facing from so many years. Let's see what that problem was. So protein's functionality is actually inherently linked to their three-dimensional structure, as we know that. So that three-dimensional structure actually is determined by their sequence of amino acids that they contain. And the process by which this protein folds into its precise and functional 3D structure, three-dimensional structure or shape, that process, as we know, as protein folding. So you may consider it as like origami. You know, origami, the art in which they fold the paper without cutting it. Same paper can be folded into maybe the ship, maybe the plane like structure or any other toy. So the number of amino acids are same. Only the sequence when it changes, the number when it changes, we are seeing a very drastic change in the shape, structure, and function of the proteins. So what is this PFP stands for? This PFP is actually protein folding paradox or protein folding problem most precisely. And the Levinthal has given that for the very first time. This protein folding problem is one of the most significant challenges in the molecular biology and in the bioinformatics. It refers to the difficulty of predicting a protein's three-dimensional structure solely from its amino acid sequence. This problem is crucial because the function of protein is directly related to its shape. And the fascinating aspect of this protein folding problem is Levinthal's paradox, named after researcher Cyrus Levinthal. This paradox highlights the seeming impossibility of the protein and its folding into its native structure in a reasonable amount of time. So let's put it in a perspective with respect to time. So for example, consider a protein which is around 100 amino acids long. So 100 amino acids long protein can have any number of conformation. So if you calculate that, that number of conformation goes to 1.58 into 10 to the power of 40. Sorry, it goes to 3 into 10 to the power of 100. And if we consider that one conformation is taking per second, so to have all these 3 to into 10 to the power of 100 conformation, it would take a particular protein that which I have written here 1.58 into 10 to the power of 40 years, which is larger than the age of the universe, which if we can convert it into the billion years, it goes to 13 billion years, the age of this. Yes. So that paradox, sorry, was first given by the Levinthal, but later on he also said that proteins actually are not taking that much time. They are folding to their native 3D functional form within seconds, or more precisely within milliseconds. And there are, of course, some proteins which are smaller. They are getting their 3D shape within microseconds. So this is a kind of opposite thing, and that's why it's called as paradox. So obviously, one conclusion can be drawn from here that proteins are not randomly folding. They are folding in a highly efficient way, and they are having a particular directed pathway rather than a random search. So on the right side diagram, it's shown that a protein tries to uh, reach to a particular level which has the minimum energy state, which is, the law of universe, the entropy. So the least energy state is the most stable and the most functional state of that protein as we consider. And also there is no scope for mistakes during this protein folding process, because a slight mistake can result in very life-threatening or fatal diseases. If proteins do not perform the folding process appropriately, or even if there are smaller mistakes, the cascade, whatever the result will be, that would be disastrous. An incorrectly folded or unraveled protein can lead to toxicity or even the cell death, and many diseases and disorders such as the sickle cell anemia or some diseases which are mentioned here like Alzheimer's, Parkinson's, mad cow. These diseases, they are the result of misfolded proteins. Okay, so here comes the important point to consider. We know a lot about proteins. We know a lot about amino acids. We know the structures of proteins. But the thing which is connecting the structure to the function, that process, we don't know anything about it. We don't know the exact mechanism of protein folding. That's what I have said. There is no one really knows specifically how protein folding is happening. How does the sequence information in these simple molecular chains they encode for the complex shape? And that is one of the most profound questions that we can ask, that biologists are trying to solve from so many years. So that brings us to the Anfinsen's hypothesis. I'm taking you through the history, through my phases. We are running with the phase one. That's mostly what happened in the past, so that we can go through the past, the present to the future. Primary structure here, as the Atkinson says, its fundamental concept in molecular biology proposed by this scientist in the early 1960s. It says that the 3D structure of this protein is actually determined solely by its amino acid sequence. And this implies that under the right condition, if a protein is unfolded or denatured, then that protein can renature under appropriate conditions. So there is no requirement of any external help. Proteins can fold themselves. Although we know there are some exceptions, I have also mentioned those in future slides, but most of them, most of the proteins, when given proper condition after denaturation, they can renature back to their native state. And that was first put forward by Anfinsen. And also for that purpose, he won Nobel Prize in 1972 for all of these findings. So here comes the three questions. Can a protein structure be predicted from its amino acid sequence? What is this folding code? And what is the actual mechanism lying behind the protein folding? So this protein folding problem, I am calling it as a PFP, has been a central challenge in the molecular biology for decades. It involves the understanding of how proteins' amino acid sequence dictates its 3D structure. So, despite significant advances for past so many decades of research, predicting the exact folding process remains complex and elusive till date. So there are so many enters in my slide. First enter starts with XRD or X-ray crystallography. We know now that's a part of syllabus for many curricula, even in the BSc also. So I'm not going in detail. But Peruz and Kendrew, they made significant progress in protein science using this newly discovered method, that is X-ray crystallography in 1960s, and they pioneered a technique where the proteins, they have grown into the crystal form, and then they bombarded it with the X-rays, and whatever outcome was formed on the film, they have superimposed those films to find out a 3D contour map of electron density. Based on those maps, using those 3D contour maps of electron density, they have manually, using ball and sticks, they have manually created the structure, 3D structure of the protein molecule, which can be seen here on the picture in the right side. And to find out the protein structure of one protein, it took them 2 decades. And they have got the 3D structure of hemoglobin and myoglobin for working about 20 years. And for this enormous discovery, they were awarded with the Nobel Prize. This is a simple explanation of how X-ray diffraction done, and not going in detail. If I have to tell you in very short, proteins are first crystallized in the step 1. Although it looks very simple in verbal form, it's not so. So once the crystal form is obtained, the crystals are passed through the X-ray, and then X-rays are diffracted because of the electrons present in the atoms of the protein. Those diffractions are captured on a receptive plate using the superimposed uh, technique. The electron density map is created, and using that map, the actual 3D structure of the protein is created. That was how the X-ray crystallography still is in the use. It's one of the best and most used method to determine the 3D structure of the protein. But nowadays we have two more techniques which are again so much popular. One comes as nuclear magnetic resonance, and one comes as cryo-electron microscopy method, in which actually there is no need to create crystals, and proteins are flash frozen, and that means frozen very fast, and then they are passed through the electron beam, and whatever pattern they are generating, that pattern is used by the computer and analyzed for its 3D structure. Okay, so here comes the researcher's problem, the agony. The method that was they were while doing the X-ray diffraction or X-ray crystallography experiment, that method that was too much time consuming, too much efforts had to be put. It was cumbersome, and to find out the structure of even a single protein, it took so many years. Sometimes people were taking it as their doctoral research, and throughout their complete doctoral research, they were even not be able to find out the structure of one single protein. So that was this problem that many scientists have faced at that time, not now. So how that problem is addressed nowadays. So let us see that. Here comes the second thing. Enter the protein data bank. So as people were getting so many new discovered structures of protein, there was a need to document, to record all these newly discovered proteins data. So here comes the protein data bank, which was formed in 1971. Protein data bank, as the name suggests, it has the database of protein structures. It is very good feature is that it is freely available to anyone without any restriction. Anyone can use it. Even I am teaching it to the students, and its database is held in the US. When it was started at the very first in 1971, it was having only 7 structures. In 1984, it was having 152 structures. In 1992, having 747 structures. And as we can see in the graph, the growth of the PDB was exponential. The number of the protein sequences, protein 3D structures that were deposited into the PDB is growing drastically. This was a completely willful idea. If you want to give your protein structure, if you want to make it publicly available, you can go and submit it to PDB. Okay, so that was the function of PDB, which is still currently running, and One of the best database used by many researchers for protein study is this. Currently, as of today, the protein data bank has about 229,000 structures, reflecting the rapid advancements in the structural biology and data sharing practices. So here comes another technique. Now since 1990s, there were two groups of people they were formed. One we call them as experimentalists or experimental biologists, which were working like you can say wet lab experiments. They were doing wet lab experiments and deducing their results. Second, with the advancement of computer technology, call them as computational techniques, people were working on the computer and trying to find out the solutions. So from 1990s, computational biologists began leveraging the computer algorithms to predict the protein structures from amino acid sequences. This marked the beginning of the competition between traditional experimentalists, those who were doing wet lab experiments, and computationalists. Experimentalists work with the precision. Those who were doing the wet lab, they were working with the precision. Their results were very precise, but it was slow. Biological experiments are slow. While those who were doing the same things on computer, they were having high speed, but actually their predictions are less reliable. They were having more errors. So those were not preferred at that time. So there was a need for better cooperation between these two, the computationalist and the traditional wet lab experiment researchers. So in order to achieve the better results, here comes a person, John Moult, and his idea, the CASP. So John Moult and the CASP, the full form of the CASP is given here, Critical Assessment of Structure Prediction. He came up with a very brilliant idea to manage the experimentalist and computational biologists together. He has made an arrangement in which he had held a conference in 1994, the first CASP conference, in which he had put forth his idea. What was his idea? You can see this part. He received and popularized the list of amino acid sequences for proteins whose structures had been recently solved. Those wet lab experimentalists, they were doing the structure identification. Those structures will be received by MALT, and the MALT will supply the amino acid chain, amino acid sequence to computationalists, those who were doing computer experiments, for example, and allow them to predict the 3D structure. So Molt was having 3D structure in his hand, for example, and those who were trying to predict the 3D structure of protein on the computer using the computer methods, computational methods, they were given only the amino acid sequence and they were allowed to predict the 3D structure. And when they come up with the 3D structure, there were separate assessors, scientists who were actually matching both the structures, those given by the wet lab experimentalists and those given by the computational experts. And they were trying to see that whether computer programs are really good or whether they are achieving the success at higher rate. If yes, we can say that yes, it is working. So it started back in 1994 and the idea worked, and the conference is taking every alternate year since then with the advanced algorithms every time. So algorithms are nothing but computer programs, you may say in short. But the issue is these computational algorithms are more like physics oriented. See, those computational people, they are not having that much knowledge of biology, so their knowledge is mostly either statistics, mathematics, and physics oriented. So their prediction initially was not that good. And whatever results they were obtaining for protein 3D structure, they were matching only up to 20 to 24% in early years. However, some methods, some people, they come up with really good outcome, and we are using it nowadays also. For example, we are using a homology modeling based on comparative studies. And till 1998, There were no proper algorithm that was proven best and could not beat any experimental biologist. But in 1998, it marked the appearance of a algorithm, software you may say, Rosetta, which was made by David Baker. That Rosetta changed the game entirely. That was having a very good percentage of accuracy. Baker also prepared one game at that time. It's called Fold It game. This folded game is also available nowadays online. That folded game is actually used by anyone in the world, can be used, and you can actually predict the 3D structure of any particular given amino acid sequence. And if your structure predicted structure is good, then that gives you the score. The success of this folded game was phenomenal. There were hundreds and thousands of people across the world. They were playing, and even one top player of that folded game is now working with the David Baker currently at his lab. That was very good thing that happened. Anyone can go and check for Google and use it, download it, and play it. Even the people who were not from biological background, they were also predicting the protein structure up to very good percentage, very good accuracy. But still, even after all this, if we consider the human prediction and computer prediction, the human prediction, that means the outcome which is coming after wet lab experiments is still better. So again, we have to wait for some more years. This is homology modeling, in which we compare any unknown sequence with all available known sequences and try to predict the 3D structure. And this is the screenshot of a Fold It game. I have also given the link, fold.it. It's available there, but unfortunately you cannot play it online. You have to download it, install on your computer, and then you can try play it. There are several things, and there is also a help file is also there. You can go and or you can ask your student if you are the teacher. So after that 1994, there comes a long wait, and in by 2016 we have got a gradual upgradation in those algorithms. As the computational techniques were developing, more and more data sharing is happening, more and more advanced computers were developed. The accuracy of protein prediction using the computer also increased gradually. By 2016, it reached 47% accuracy, whatever developed on computer with the actual one. By 2018, it has reached up to 70%. But there was still one problem. How much accurate structure can we consider as okay? Can we consider as reliable? So that threshold was designed by John Molt himself. He said at least 90% accuracy should be there between computer derived structure and the actual X-ray diffraction derived structure or whatever, something like that. So here comes another AI tool that is actually given by the Google. It's called DeepMind. DeepMind is the branch of that Google who was working on AI, still working, and they have put forth a new era. Here begins our phase two, which is AI era, which started several years ago only. So Google's DeepMind developed a neural network. Those who don't know computer things, for them I have simplified it as there are neurons in our brain which are connected with other so many neurons, and then there is the transfer of those signals. Same way, there are various layers of neurons that can be made in the computer algorithm, and they can interact with each other just like the neurons of our brain interact. That network is called as neural network. You might have heard because AI is very popular nowadays. It's called neural networks. So neural networks have ability to learn and understand. And our AI, if you are aware about the chat GPT and etc., these are based on these neural networks. So with the advancement of these neural networks, we have better chances of predicting the protein structure, 3D structure. So here are there are two neural networks I have shown here. Not going in that technical part, but if you are given an input such as a sequence of amino acids, it is processed through the multiple layers of informational nodes, and that those nodes are called neurons. Then each neuron calculate whether to send the signal onto the next neuron in the next layer. And after so many these evaluations, repetitions, algorithm gives out an output. For example, a protein tertiary structure. In our case, in biological case, same is true for other cases also. The more number of layers we have, the more intricate number of or more higher number of calculation can be performed by that neural network. So earlier days there were only two layers of neural network, so number of calculations were less, the time taken was so high, and the accuracy was less. Nowadays we can have very large number of layers in the neural network, so that the number of calculations, number of interactions of nodes is increased, and because of which we are getting more precise and accurate structure. So that part we call it as deep learning. Jinbok Zhu, who brought this technique to protein folding. So obviously that was the computer part. Somebody has to take it to biology. So this is the person who does that. He used the mathematical object called matrix to represent which amino acids were close together in space, then fed the data into the convolutional network as an image. An algorithm took it from there and derived the structures. So Google DeepMind is a new player now we are seeing in the picture. One important thing I would like to tell you, any neural network or any AI that you see here used, that is trained on some already existing data. If there is no training or the data available is less, then the prediction will be less accurate or there will be no prediction. So in order to train the Google's DeepMind algorithm called as convolutional neural network CNN, we were already having around 140,000 protein structures. Obviously, they were available in the PDB for free. Those were used by Google's algorithm to train. Train means what? They were understood by the neural network, and then once they understood, they can now predict. The algorithm was, sorry, this algorithm was named as AlphaFold 2. Sorry, AlphaFold 2, the Google's AI. It's a prediction engine. For each sequence, amino acid sequence we give, it gives out the prediction close to experimental structures, and which is presented in the paper, which is also freely available in the Nature magazine. And Google's DeepMind submitted all their predictions to the CASP conference. As I told you, CASP conference takes place every alternate year. So CASP conference, they give out like challenges. These are the amino acid sequences. Try your algorithms and come up with the best possible 3D structure prediction. So that was actually given in the CASP 2020, not a long ago, only 4 years ago. We have seen that. CNN actually based on two kinds of neural networks. Earlier, it was called CNN. Now it is based on transformer. One very good advancement that has been done is that in the CNN, one kind of node is interacting only with few other nodes. So there are less interaction. You can consider a neuron. Our brain neuron is connecting with, say, for example, 10, 15, for example, 10, 15 neurons. While the new algorithm which was developed, transformer, one neuron or node is actually interacting with all other nodes, which is actually a better thing. And because of this advancement, more better prediction of the structure was possible. Okay. So here Google has given their structures to the CASP conference, or John Mould, independent scientist, team of scientists, assessors. They have obtained those structures from Google's DeepMind AlphaFold, and they were already having the 3D structure which were developed by wet lab experiments, and then they compared it. And here came the earthquake. In the CASP 2020 conference, which was witnessing the thing, the structures were so accurate. The structures were so accurate, which were given by the computational methods, that they have reached a score of even 100%. Okay, so during that CASP conference, assessors scored the thing, a perfect score in which the model and reality match even atom by atom is 100, and anything above 90 would be considered as reliable or accurate. And most of the AlphaFold's structures hit surpassed 90 marks. So it's a score given from 0 to 100. Anything uh, 90 or more than 90 is considered as correct or good or reliable. And most of the structure which were given by this prediction program, the AI, was hitting 90 marks. And some even reached 100 marks, 100 score. That is 100% similar with the those developed by wet lab experiments. That was a moment of eureka we had witnessed in few years ago. So this AlphaFold, what happened? Let us see what were the issues, what happened later. This DeepMind AlphaFold had solved structure prediction part of the protein folding problem. We can say like that. It had solved. Like it could accurately predict the structure of the protein from its amino acid sequence. That was the outcome. We were looking at it. Then there came this moment of awe and shock, because computational biologists who had been trying to solve this problem for some decades, they found the moment of awe and shock, because we were looking at this problem as one of the very uh, impossible kind of problem. We are not able to deduct the protein structure in so short amount of time. What they were doing for months and years can now be done in days or even overnight. So finding the structure of a protein only using amino acid structure is now the matter of only few minutes. If time permits, I am going to show you live. I have already opened a website which is AlphaFold 2. And good thing again, most of the bioinformatic tools are free. That's one of the very good thing humans are doing it. These tasks, they were previously taking months and years. Now it's happening in what with just few clicks of buttons. For structural biologists, AlphaFold to revolutionize their ability to hypothesize the protein function. What is the next step? Now we know the protein structure. Next step is to find out their function. People, wet lab scientists, they were first have to identify the structure and then propose their function. Now, as the structures can be done in very short amount of time, now they can hypothesize their function and only work for checking the function. Okay, validation of their hypothesis. Now they could generate hypothesis within minutes. So AlphaFold 2, just I'm giving you an example, real example. Prediction of Cas14 target T1049. PDB means the protein data bank ID 654F, which is shown with the blue color, and the green color is the structure of the protein predicted by computational method, by AI, for in simple language. Okay, so this blue color and green color, you can see it's very superimposed. You can see at the bottom the score comes 0.93. That means 93%, which can be considered as reliable. Another example, which has been, there are so many examples, I have put only two. The Cas14 target T1056 PDB ID is given, and you can see the green and blue. Those are very much superimposed. And AlphaFold has accurately found the side chains also, even though it does not explicitly predict the zinc ion. Okay, so that was the power of this AlphaFold 2. So this problem is now solved. So what next? Finding the protein 3D structure is now a matter of minutes. I can do it, you can do it, anyone can do it. So what is next? So currently, with AlphaFold 2's remarkable capabilities, scientists and researchers have adopted a new workflow for their projects. Any new scientists, they were initiating their new study, they first utilize AlphaFold 2 to predict the structure of a specific protein. And these predictions, they provide valuable starting point for their further research or process. However, it is still essential to validate these predictions experimentally. So I'm not saying that because of this computational power, we are ignoring XRD or other conventional methods of protein structure determination, XRD, NMR, cryo-EM. Yes, they are still important. Whatever we are getting using the computer, at least for some next years, until we become very sure, we have to validate those through the wet lab experiments. But at least we know what to do. We know where to go. We know the destination. So once you know the destination, you know exactly how to do or how to go. There comes the shift. This nuclear pore complex, which was considered as one of the very complicated protein structure, its 3D discovery was influential due to its complex nature. AlphaFold 2 has predicted protein structures that have been used to study diseases and create new tools for drug discovery. However, this is just the beginning of what AI can achieve in the field of protein science. The future holds even more exciting possibilities as the AI continues to evolve and improve. Just like humans, just like animals, just like living organisms, AI also improves. Here enters a new thing, that is novel molecule designing. So, can we design molecules which are not present in the nature? That becomes our next frontier. That is becomes the challenge for scientists. Scientists start seeing beyond the nature, beyond those proteins which are available naturally. They are now utilizing AI not only to discern the structures of and functions of the known proteins, but also scientists are using AI to design entirely new ones, those proteins which are not existing in the nature. So this technique is pivotal for designing and developing innovative therapies. It has the application in medical field. The ability to create custom proteins could lead to groundbreaking treatments for various diseases and the advancement of biotechnology. At this point, when other companies they also saw the outcome of this Google's DeepMind, many rival companies they entered. They entered into the scene and they have tried to develop. One such new algorithm that came into existence is Rosetta Fold, given by the David Baker, US. The full form of the Rosetta is given here. Meta has also entered in the same field. So far, the AlphaFold 2 is still better than all its alternative. So right now, biologists are still confined studying the proteins already invented by nature. But can we design ligands or small molecules that could bind to specific receptor or other protein molecule? Example is given here, receptor protein of the virus or bacteria. Can we design such a molecule using AI? Or can we design something that harness the sunlight and break down plastic? Can we design those molecules which can be used to uh, solve the problems which are we are facing globally? So let us see. Here comes another thing. Enter the reverse engineering. Uh, as the name suggests, it's a self-explanatory. Here we have amino acid, then we have folded structure, and then we have final 3D structure. So instead of going from this to this, can we go from this to this? Can we go from structure to amino acid sequence? So let us see what is reverse engineering. Yes, it is possible now. More shapes we can deduce, and that means more functions we can have. The protein designing nowadays is using inverse protein folding problem, which is basically exactly reverse of protein structure prediction. You were predicting the structure from amino acid. Now, instead of predicting the structure, design a new protein itself, which can bind to a particular receptor, and then try to find out a amino acid sequence which can produce that amino acid. So rather than feed an amino acid sequence to a deep learning algorithm and ask it to give out some protein structure, now scientists are doing, they are doing what? They are designing a protein, and then they are feeding that protein to the AI and asking the AI to generate amino acid sequence which can produce such a protein. After transcription translation, we know that. Then using these amino acid sequence, designer proteins are built in the lab. How it's done? We know the structure of the protein. We can deduce the amino acid sequence which can give out that protein after translation. We know the amino acid sequence, then we get the complementary RNA sequence, then we get the complementary DNA sequence. Once we get the DNA sequence, just throw that DNA sequence to the DNA synthesizer. DNA synthesizer will synthesize the DNA, use the genetic engineering to transfer the DNA, the synthesized DNA into the bacterium, make the bacteria more unique. And once the bacteria is there, having that foreign gene, it will produce that protein, harvest that protein, and use it. One very good example I can quote here is for venom. For example, venom. Antibodies against snake venom. At present, we are using horses to produce anti-venom, antibodies against the venom. So that's unethical also, but time consuming also. If you know the monoclonal antibody, that process. So instead of that, just like we are producing insulin using bacteria, can't we produce other proteins by the same process? Okay, yes, we can do that. Just a minute. Sorry. Here some of the examples, real world examples, which were actually created by this reverse engineering. They are put here. HIV-1 protease inhibitor, which is inhibiting HIV-1 protease, insulin analogs, lactase enzymes, green fluorescent protein. But one example I have put here, an animation you can see here. In this animation, actually, a new algorithm is used called RF diffusion, called Rosetta fold diffusion, which is actually given a prompt to generate a new protein molecule which can bind to this already existing blue colored receptor molecule. Okay, so I already have this receptor molecule. I know this receptor is actually, for example, it's a receptor from virus surface and is actually the binding to the human cells. So instead of what I do, I take that structure of the receptor, I will put it into this AI and I will put a prompt to produce a certain 3D protein which will bind to that receptor. Now it is possible. A new algorithm has been developed called Rosetta fold diffusion. And very good thing again, it is free to use. Anyone can use it. And what it was taking months and years to develop find out some natural remedies over the diseases now can be done within days or even overnight. Okay. So is this problem, the protein folding problem, really solved? So if we can design new sequence that fold up to a new expected structures, then that shows we understand quite a lot about protein folding. And there were scientists, that's a 6-step process. It starts from denial and finally ends it into the acceptance. So nearly all of the biologists, they have gone from denial stage to acceptance stage of the use of this AlphaFold. AlphaFold success has been undeniably shifted biologists' attitudes towards AI. And experimental biologists, they started believing the outcome of this AI, and not only they have started accepting blankly. Yes, they were backed up by wet lab experiments also. So here comes the most amazing part of my talk, this slide. It took more than 50 years to deduce the structure of 229,000 proteins. More than 50 years. Earlier, it took, we have seen 20 years to deduce the structure of hemoglobin and myoglobin. But in only 2022, DeepMind's AlphaFold released the predicted structure of 218 million proteins. 218 million proteins. Nearly all proteins known to the mankind. All their structures were released by DeepMind, AlphaFold. All those are available in Protein Data Bank in a separate category, just like they are called as predicted by computational techniques. And all those who were predicted by wet lab, they are still have their separate category. But at least you have them. Okay, that's too much positivity about this use of AI and etc. etc. Let's talk about some drawbacks too. I need not to tell you everything which is good. I'm going to tell you what were the drawbacks. So first, very important cons is minimum 90 score is still not perfect. We know that. Even one amino acid change can cause a serious disease. We are very well aware of that. So 90% accuracy is still way behind what we can trust blindly. So 100% is a must thing. But as I said, we are all evolving. AI is still evolving. So in the very near future, we possibly are getting 100% very soon. Proteins. Another drawback of this AI deduced structure is that proteins they work in consortia. We know that there is not a single protein in a cell. Sometimes protein need the help of surroundings, such as certain ions, salts, metals, other proteins, chaperones, to fold them properly. So AlphaFold, that AI still lacks this ability of prediction. So we know that there are some proteins they fold in a proper environment. Okay, inside the cell. Maybe outside the cell they may not fold as intended. That thing is still lacking in AlphaFold's algorithm. This is one another drawback. A few more drawbacks I'm going to tell you. There is a kind of protein they are called shape-shifting proteins, or fold-switching proteins, and they change their shape upon interaction uh, uh, with other molecules. That change ranges from small to large scale. For example, some protein they bind with the DNA during DNA replication and they change their shape slightly. Enzymes we know are, for example, flexible. They may change their active site shape slightly while during the reaction. AlphaFold fails here. The prediction fails here. But the reason of failure is obvious. As I told you, to train AI, we need a considerable amount of data set. We have hundreds of thousands of data set of static proteins. That those proteins who are simple, who are monomeric mostly, single structure proteins. We have a lot of those proteins to study. But these proteins which are shape shifting or which requires a complex environment or which requires complex interaction to fold, etc. Their database is limited. I have mentioned here there are around only 100 such examples. So 100 examples are not enough to train the AI model and use it for prediction. As we go through the time, we will be having more such protein produced, structure produced by wet labs, and then we will be having more and more training of the AI. And in the future, possibly, we are going to reduce structure of those proteins also. So here in the animation, I have shown the example of protein shape shifting proteins. But still, there is a hope as AI is evolving. In September 2023, Alpha Missense, another algorithm was developed which actually predicts the possible mutation. It can't show the exact change in the structure of the protein due to mutation, but It informs the user if the mutation might turn the protein into pathogenic or introduce some dysfunction. All proteins are not harmful. We have silent mutation, nonsense mutations. So this mutation actually that AI can predict whether it will drastically affect the protein or not. So we have reached up to that much path in this journey. Also, we know that cells are very complex. There are n number of proteins, n number of other chemicals. Even there are thousands of different proteins present in a single cell, and they proteins interact with other proteins, other molecules, etc. This interaction is a very complex thing. And still, we are not very much aware of all these kinds of interactions. So, AI has limited knowledge in this case also. So, again, AI has limitation in this domain to find out the protein protein interaction or how the proteins will work, behave, newly designed protein will work, behave inside the cell. That can be done only through wet lab experiments. So in order to solve that problem comes a new algorithm called Rosetta Fold All Atom and another algorithm that came as AlphaFold's next version, AlphaFold 3. Both these algorithms they were launched very recently in the past year only 2024. It enables AI to predict the structures of protein which are bound to each other or to the DNA or to RNA or other small molecules like any ligand molecule. So you see, the AI is evolving, AI is growing. Now we have this information how the protein will behave in bound state. The testing of this AI is still ongoing, and so far out of these both algorithms, the AlphaFold 3 is prevailing in accuracy. And also AI is less good in drug docking since I told you the database is limited. Therefore, drug docking studies is still in the evolving phase. This is AlphaFold 3's prediction, which is done in the real life example. AlphaFold 3 predicts the structure of molecular complexes such as this enzyme found in the plant damaging fungus. And in this model, the protein, which is shown with the blue color, is linked with the sugar, with a sugar molecule, which are shown with the yellow color, and also an iron molecule, which is shown in the yellow sphere. So that which was earlier not there, the interaction with other molecules, now it is there. Obviously, it is going to help scientists in a new way. So Rubenstein is the researcher who is currently working on cancer research. She is trying to work on kinases to find out the cure for cancer. It took her 2.5 years to develop the 3D coordinates, I mean, the 3D structure, exact 3D structure, atom by atom of this enzyme. And recently, after the launch of this AlphaFold 2, she used AlphaFold 2, and very within few days, she has reduced the structures, which are more than 80% accurate. That's another example. So here comes the phase 4, the last phase of this complete talk. That's conclusion and what is the future of this. Conclusion comes for many biologists, AI structure prediction algorithm was the breakthrough they had been waiting for years. Of course, 218 million protein structures were deduced only within a year, for which we were working from lot of decades. Okay, now we just have to verify them. Okay, Google's DeepMind played a pivotal role in this case. CASP, which is a conference held by John Molt, raised the platform. He is the person who first put forth all this, bring the people from computational domain, biological domain together, made the connections and made this happen. Many competitors are now entering in this field. Many new algos are being developed, and which makes the scope for research in this field. Uh, Bioinformatics is now the field where you have the knowledge of biology, computer, and then there is a good scope. Even for the students, your students may opt for this field. So I am re-questioning the thing again. Is the PAP really solved? So AlphaFold 2 can recognize pattern in how a given amino acid sequence might fold up based on its analysis of hundreds of thousands of protein structures. but it cannot tell scientists anything about the protein folding process. So if you remember, I have posed three questions earlier during the initial part of my talk. Uh, are we going to predict the structures using amino acid sequence using computers? Yes, it happened. The last part, are we going to know the mechanism actually how the proteins are folding? That part is still yet to be discovered. We don't know exactly how it is happening, how the protein is folding in biological condition. That part is yet to be unraveled. So conclusion you can take home is, if you can cure the cancer with this new AI technology, do you care about how it really works? This is one kind of view one can ask, one can consider. It is a question that we are going to wrestle with for years to come. And 70 years ago, we were actually thinking that proteins are only some gelatinous substances. And see what happened in the 70 years, how much knowledge we have gathered. So obviously, there is a scope. Uh, One of the last slides, I just want to read it. If many researchers decide to give up on understanding nature's processes, then artificial intelligence will not just have changed science, it will have changed the scientists too. This is a food for thought. And with this, I end my presentation. Thank you. Thank you all. Over to you, Tejas.
So, thank you, dear Kamlesh sir. I request participants, if you have any queries, you can share with him. Yes, participants, are you there? One of the participants has raised the hand. I request
Good evening,
Dr.
sir.
Mr.
Pratap
Pratap.
Davare.
Am I
Yes,
audible, sir?
Yes, you
yes,
are
sir.
audible, sir. Please go ahead.
Sir, I want to ask one question.
Yes,
Sir, uh, what is correlation of docking score and its activity?
sorry.
Is there any correlation between docking score and its uh, pharmacological activity?
Yes, sir, there is the correlation. We know that docking you want in which context?
Sir,
From
I am medicinal
pharmacological.
chemist. So we are
Yes.
performing docking of structures and we are
Okay.
predicting activities of that structures.
Okay.
So what generally happens, we are getting good docking score, but sometimes it fails to show the pharmacological activity as well.
Okay. On computer, you are getting the structure correctly.
Yes.
Okay. So that's what I said. When that we are doing something on computer, it obviously may not work exactly as we expect because biological systems are very complex. You know, when that same kind of thing is going to be happen in the biological system, as we know, because of other interactions, whatever we are expecting on computational methods may not happen exactly. That's why Rosetta Fold all atom is going to work on, and very soon you are going to be testing that thing. We will be able to test that thing also.
Sir, what should be the minimum score that should be considered docking score?
For docking software,
Yes,
I can tell you from AI point of view,
okay.
its minimum should be, as I said, 90 must be there.
19.
90, 90.
Okay.
See, anything below 100 is always not reliable. You know,
Okay.
even you get 100 score in the on the computational methods may not work in the biological system. So our whatever computational things we are doing, they must be tested in vivo, and if they are not working, we must find some other way.
Sir, as a you are an entrepreneur, I want to know
Yes.
what your company is doing presently.
Yes, we are providing bioinformatics services to researchers, students.
So, are you providing simulation study also?
Yes, we can do that. We have a team with us. We have experts from different areas, and we can connect you with them. Yes.
Okay. Thank you, sir. It was very
Thank you.
informative.
Thank you. Thank you.
Thank you.
Yes, anyone else from the audience?
Hello, sir. Good evening.
Yes.
The lecture was very nice, very,
Thank
very
you.
enlightening, and very nicely you explained all the developments and evolutions in protein structure understanding.
Yes.
It was very, yeah, very nice.
Thank you, ma'am.
Because many of us belong to life science and this bioinformatic part and the tools that are available to resolve the
Mhm.
things, we are not aware exactly. Now we came
Yeah.
to know that these tools are available in open domain and can be used by anyone. So
Yes, ma'am.
yeah, definitely it will be useful to all of us. Sir, one
Yes. Yes.
question I wanted to ask you about recombinant proteins. uh,
Okay.
as you said that synthetic, if you know, I mean reverse engineering, if you know the protein
Haha.
structure, I mean normally we go by that route. If you know the structure, then we go for the amino acids, from amino acids gene data, and then we try to clone it and then we try to get it. But
Yeah.
yeah, but first question will be, when these recombinant proteins are produced, many times they don't fold in a way like the native protein.
Hmm.
For most, I mean many proteins we have experienced that they don't fold in the same way. Then we have to put chaperones and other things also to be cloned,
Yeah.
because they don't, yeah, and because they are not folded in the proper way, they are non-functional, as you explained in the beginning, and say they are not
Yeah.
soluble, and
Mhm.
then it is a toxicity for the cell. That,
Okay.
then in that thing, how these things are resolved. And second thing is, when we talk about producing these proteins by this
Mhm.
method, then synthetic
Mhm.
peptides, if we have amino acid data, then we can go for, I mean, there are many resin based methods where synthetic peptides can be produced using amino acids.
Mhm.
So, are these methods, I mean, equally suitable, or we have to go by cloning method only?
Okay. Yes. You asked first question. Just like the sir asked the same question. What we are seeing on computer on screen is not actually working in the biological system.
Yes,
That's
yes.
one point I have already explained. I have put the diagram. The cellular systems are so complex. Sometimes we know that proteins need a post-translational modifications. Na? We are not considering that part. So maybe the post-translational modification is one thing. Maybe protein that we are putting in the cell may get aggregated or may not reach to actual its substrate or destination. Sometimes
Right.
the expression system that we are using
Using is not correct.
can be causing problems, so that can be changed. One thing, and again, if the protein requires, as I said, extra components for their folding, salts, ions, chaperones, then
Right.
definitely you have to put that part in your consideration. And second thing you have said, although resin based
About it.
thing is right, yes, it's time saving, but more proven technique is cloning based technique that gives you more accurate result because that comes from biological source.
Biological source. Acha.
Yes,
So that
so that
method
will be more accurate.
more accurate.
Yes.
Okay, sir. Yeah. Thank you.
Okay. I just want to show one thing. If no one is asking anything else, Sandeep sir, just 1 minute.
Yes, yes, we can have.
Ha, ha. Yeah, I wanted to show this. Um, this you go on Google and write AlphaFold, and this AlphaFold 2 will open up. And there is a course they are teaching to you. You can go through all these articles step by step. And again, you have protein sequence with you. There is the workflow given by them. You can use AlphaFold here. If you have so many number of proteins, then you can even download it on your computer and run it on your PC. But for that purpose, very high configuration computer is needed. So it's better to work online. And then you go for Google search AlphaFold Colab. Then you come to this point. I forgot, actually, I wanted to show you how it works because it takes some 5-10 minutes. And there, if you are you seeing the screen? Screen visible, no?
Yes, sir. Yes, sir. Yes, it is visible.
So here, yes. So here there is a amino acid sequence, single letter amino code you can put here. For monomeric protein, if you feel there is a multimeric protein, you can put multiple sequences here. And you go into the runtime on top and run all. And that will take hardly 10 to 15 minutes and you will get your protein 3D structure developed here. So how fast protein structure determination you can see has possible because of the AI. Earlier it was taking years and years. So that's what I wanted to show you. Thank you.
Sir, I have one more question.
Yes.
Sir, which point should be considered for while selecting the protein structure for docking?
Which point to consider for?
Selection of the protein structure for docking study.
For
And
docking.
there are so many PDBs available in data bank.
Achha.
So of them, which structure we should consider for our docking study?
Okay. You wanted to select the ligand molecule from the PDB?
No, ligand co-crystallize with protein structure.
Okay. So either you should go for several things. First is if you are searching the molecule from the PDB, search for the resolution and the quality of that molecule. The ideal resolution should be 2.5 angstrom and better than that. And search relevance of that protein structure from the biological context. Also check if the ligands are needed for its functioning or not. Okay. Also search for protein flexibility. These points you may consider while choosing the molecule.
Okay, thank you, sir.
Yes.
Well, if there is no questions anymore, let us thank Kamlesh sir for such a wonderful lecture. It was really a new thing that many of us haven't came across. So very nice way to present the new happenings in the era of AI and biotechnology. Thank you, Kamlesh, for joining us. Thank you very
Thank you,
much
sir.
from on behalf of the department as well as on behalf of all the participants. Thank you and see you.
Thank you,
Good
sir.
night.
Okay, sir. Thank you.
Sandeep sir, can we go ahead with Chandrasekhar Patil sir?
Yes. Chandrasekhar sir, are you there?
Yes,
Hello?
I'm there. Yeah. Am I audible? Yeah.
Yes, you are
Yes,
audible.
sir, you are audible.
You are now presenter. You can share your screen, and yes, you are also visible now by video.
Yeah.
Long after a very long time, sir, to have you with us and
Yeah, it's been, I mean, last time I remember during the pandemic, we had
right in the webinar.
symposium.
Yes, webinar.
So yeah, almost like 5 years maybe. Yeah.
Right.
So yeah, no, it's always good to be here. So and good in connection with the college, like how the R.C. Patel College is progressing. We feel like really happy to see the way department is heading under both of your leadership. I mean, when we were the student, then Dr. Ullas Patil sir was there, and then now you took over in a like successive manner. So that's really great to see
Yes, sir.
from our side because we have seen the college progressing. I was at the Shirpur College like very first time as a part of contested for Louis Pasteur competition. So
Yes.
Right, sir. Right,
back
right.
yeah. So and that was very like impressive the way that time that kind of competition was going on in the field and for our area of like I mean I feel that time I felt like okay Shirpur College is thinking like in the future and that's what I think like helps the students as well. So
Right, sir.
yeah.
Well, so we'll start. Once again, welcome back, ladies and gentlemen. We have with us our second resource person, Dr. Chandrasekhar Patil, and I take pride in saying that he's the product of KBC NNU. Uh, uh, was my colleague during my PhD days, and he is now working in the university where our great Ananda Mohan Chakravarti worked and where he designed the world famous Pseudomonas tutida. All of us maybe probably familiar with that. So sir is working in that prestigious University of US, University of Illinois. So I welcome Chandrasekhar Patil sir on behalf of the Department of Microbiology and Biotechnology, RCPL College Shirpur, and on behalf of all the participants of this FDP. So, sir, this FDP is going on from last 5 days. This is the fifth day actually, and we had very stimulating discussions from different scientists having different expertise. And we have very vibrant participants. They do ask a lot of interesting questions. And I hope this session will be also engaging and stimulating. So before we formally begin this talk, I request Tejas to please introduce our today's guest to our participants. Over to you, Tejas.
So I'm introducing Dr. Chandrasekhar.
No problem, sir. Please go ahead.
So I feel really very happy to introduce our resource person and one of my good friend for the second session, Dr. Chandrasekhar Patil. Dr. Chandrasekhar is a research assistant professor at the Department of Ophthalmology and Visual Sciences at College of Medicine, University of Illinois, Chicago since 2024. He has previously worked as a research scientist at the University of Perpignan via Domitia, France. He was a postdoc fellow at the Ben Gurion University in Israel. He has received AEVR Emerging Vision Scientist Award in 2024. He has received a prestigious Marie Curie Fellowship in the year of 2017. He has also received Young Scientist Award by ACRB in 2015. His research interests include ocular infections, neurodegenerative diseases, large data sets in public health. He has published around 90 articles with 3,500 plus citations. So with this, I conclude and I welcome Dr. Chandrasekhar to share his expertise on biotechnology focused on artificial intelligence for biotechnology research. So I once again welcome you, Dr. Chandrasekhar, and over to you now, please.
Yeah, thank you again, Dr. Sandeep and sir and Dr. Ravindra sir for the kind introduction. It's again, I just repeat that it's again nice to be here among the people here. It feels like homely when we talk for our institutes, and that's it's also like the we feel like it's like the giving back because we have learned so much from there for using the resources. Uh, from all the institutes in our area, and that like whatever we can in our own capacity we can share it back. That's like the great feeling, and I wish the people here, like the especially students, they will do the same in the future as well. So with that I would like to start about my talk. So I mean, I just attended, I just joined 5 minutes before, so I heard the last conversation from Dr. Kamlesh's presentation, and I get that maybe there might be some part might be overlapping. This is like not my core area, AI in biotechnology research, but I thought like the faculty development programs are like kind of important for the future trainees, and I thought to share what I know, and then also what in which direction I'm working as a researcher to contributing to the AI. That's where I see that this might be helpful because I'm from the generation when the BTI was just introduced, and we used to hear about the how the BTI is gonna revolutionize the biotechnology, and now BTI is like day to day in our day to day life. Agriculture in the pharmaceutical in agriculture practices, it is like the normally getting used, and that's how I feel that now this in this era of AI, right now this is in the very developmental phase, but maybe 20 years from now we all will be like very familiar. We all gonna be very familiar with this AI. So that's the question. So do you see my full screen here right now?
Yes, sir, it is visible.
Okay, thank you. To start with, I was looking at like what is ATAL Academy like is want to learn, or what is their vision, and that was very inspiring. Like how with their these kind of programs gonna help the faculty as well as the students for to achieve their like the lifelong learning goals through higher education, and that's gonna provide them access to knowledge and give them equity in the knowledge generation, as well as they're going to improve their quality of life through the developed knowledge. So I'm glad to say that I personally have like has like helped improved, get like lot of help from this faculty develop programs through my mentors. So I would like to say I was the passive receiver. So earlier these days when I was at the North Maharashtra University during the PhD time, my PhD mentor, Dr. Satish Patil sir, he used to attend faculty development programs, and he used to share his experiences with us. So I remember one of his talks from Dr. Dhanad sir. So I think he attended at Aurangabad University, and he was sharing that for the researchers particularly, how uh, like the researchers kind of make a contribution for the improvement of our economy overall. So like the one example he gave that, so Dr. Dhanad gave to their class, like when we publish any scientific paper, that means we are generating new knowledge, and we are kind of adding our own contribution to this global ecosystem, which is based on the knowledge, and that knowledge contribution directly leads to the economy, which we can see from these recent papers where it is proven with the long-term studies that the more the knowledge we generate through our research activities, that eventually contributes in the development of our countries. So that's where I think that motivated us a lot, like publishing any single like paper is basically you're adding a making a adding a dot to this whole sea of information, and don't underestimate that power. So over the time it's gonna grow. We can see like over the years the citations we receive, that means that many people have used the knowledge we have generated, and this is important for the students because at this time during the postgraduate studies they are involved in the projects, and that project ideas, whatever data they generate, they at least try to publish that data, so that way what information they brought out through their expertise is going to be the available for future generations, and that every small observation, each small data which is based on repetitive observations is very important and it adds up to the development of your country. So in this case also, I would like to tell that this is continue versus continual learning. So when we are like as a student from writing from start from our early age to the until we to our highest degree, so we continuously in that education process, so we learn every year some new, we add new layers of knowledge to our understanding. But when we are like go out of our actual college settings in our day-to-day life, then that learning becomes continual. So like we learn throughout the life, so we have to focus on that continual part as well. So it's not like we are done with the degree, that means now we don't need to learn anything and we know everything. So every day, each and every day, there are new developments, and how those developments are associated with your knowledge, your existing expertise, and how you can improve your skill set based on that knowledge is very important. And I think that faculty development programs, they really help uh, in making that continual learning available for the students through the improved faculty skills. And I'm really want to congratulate R.C. Patel College for their efforts in contributing to these efforts. So thank you, organizers, for doing this initiative. I know it's like if we compare the gain, I think all the attendees, including me, are more gaining more through these programs than the time you people are spending, even at the evening time. So that's really worth of work and encouragement for the all others to learn from it. So before starting, I would just like to give a brief overview, like how I will, what is included in my talk. So I would briefly introduce the AI. I will not go more in details because I believe the earlier session might have already covered some of the part, and how the AI is like getting developed in the context of biotechnology research, how artificial intelligence is revolutionizing in our life, and most most importantly, how do we integrate these models in our research studies? And I will briefly discuss the opportunities and challenges associated with the AI in biotechnology. So just to begin with, I would like to put a disclaimer that there is no conflict of interest. Some of the concepts and data are like yet to be published, and I acknowledge the AI tools and the information I used in this presentation that is coming from public domain, and I do not claim any right to that information. So when we ask like this question, like how we think that the AI and biotech are already reshaping our world, earlier I thought to take a poll, but I think anyway, that's not a good idea now. Like, so just to participants, you can, as simple as you can put in the chat box, like how do you think that the AI and biotech are already reshaping the world? So I think in our day-to-day life, we might be seeing all around the new information, like this is the new AI tool, this is in the biotechnology, what is happening. But when as a student or as a faculty, I think that also makes us think how we can connect these things and how we can put those things together so that can the field can grow even further. And as you already, all of you might have know that this everything is coming in with the prefix AI is all around us during in the news or even in the applications, biggest economic developments. If we look at the economy of the AI field, specifically in the biotechnology, it is like significantly growing, predicted to grow over the next few years. And I think beyond that, and North America is currently leading, but India is also not really behind of anything, and they have already committed like the huge amount of money to put into this business which can connect AI and biotechnology. So I think the most important difference I see compared to when we started learning biotechnology versus now. So like that time biotechnology was mostly like in the conceptual phase where we had few applications, and then we started learning how biotechnology might be helping us. But now it has like the stronger roots in terms of products, and now people can see the real impact. And for the future trainees, that could be the opportunity uh, to start their career by developing new and new applications. And there is also right time with the economic support that the government is willing to support you through your innovative ideas. When I think about how, like for us, like how India is preparing for AI in biotechnology, so these two recent news caught my attention. Like recently, India has kind of accepted, it's like the new policy which they call Bio E3 policy. So Bio E3 is basically economy, environment, and employment. So I think biotechnology is one of the pillar which can, through the sustainable development, can generate the economy, can protect the environment, and also provide an employment, which was recent, like previously was one of kind of not very much developed field. But I think with the increasing applications, that employment through biotechnology is also now one of the main goal of policy. So now there is, and also like the recent visit of the Microsoft CEO Satya Nadella, he announced like that Microsoft is going to invest almost like $3 billion. That's a huge investment for India, and they are planning to generate almost 1 crore of trained employment skill set by the year of 2030. So that also gives you the sense of opportunity, like how the global companies are looking at India, looking at their manpower and willing to employ those skills and investing the money that can help this effort going even faster. So these two news, I think they are good enough and convincing to think that now there is a real environment for to set up the new level of generation that can be contributing for further development of AI in biotechnology. So this, but I mean, AI, you might already know that AI is like not a new word, but it's like it was basically coined in 1956 at the Dallas conference by John McCarthy, who was the computer scientist. So, I mean, there might be debates over whether it is artificial or real. Artificial in terms of because the knowledge is already developed and now the machine is learning that knowledge to make some new interpretation. So in terms of that, it is basically taking the artificial information which was generated by the human brain. I mean, we always need to remember that humans are like the knowledge generators, while the machines are knowledge consumers. So they consume the knowledge and then they make the interpretation, they understand the pattern, and they make the new conclusion that can help faster our life or make our life convenient. So when about the artificial intelligence, it's we call it, so we can imagine the era of like calculators. So how the calculators revolutionized the calculations for day-to-day life. So even before computers, calculators, they just simply help to make the calculation faster. So we never imagine that just by putting the numbers, those what our brain can make, that small instrument can make it. And then the next generation is like now the smartphones, like how the smartphones are also making things very convenient for our day-to-day life. Just through simple apps, we can know the health of the plant, just scanning the photograph. How it works, it's basically that information is already used up to train that application, and that is helping us now. So earlier we used to have the experts for particular subjects, and then we need the library, but now the machine learning, we can put all information all together, and that can give us the right away the what could be the best way. But also, like, here is like best in the sense not that's the final way, because there is always human aspect to that. Like the humans can always uh, need to be rethink about that, and that I will discuss more in detail in the coming slide. So some of you may have seen this, like the small wonder. I would like to bring this because that's how when I was a kid, I came across like the robots, like how that was one of the TV serials during the 90s based on the American TV serial that the computer scientist he develops a one small robot, like the humanoid robot that was meant to be assist them for their day-to-day life to make their tasks simpler. That was completely based on like the fiction. That was the science fiction, and that was one of the very popular trend, which can give us the sense of future. Oh, how in the future robots might be helpful for the humans. And now today, if we see that these things are become realistic, so within the 30 years of time span, uh, we do see that the robots are now the reality in the world. They can do very delicate things, and they can also help in our day-to-day life. Like this Tesla robot is one of the would be the very appropriate uh, example for this. But when we want to think about the next 20 years, I mean, I think I can give you the very simple sense of how could be the next, it is going to be development. And you might aware of this, like the recent movie where they show that how the humanoid robot was very similar to the humans, and people can also can just imagine the difference between humans and robots, and can find them as their partner. So I think maybe down the 20 years down from here, you might be talking about, oh, we used to think that watch that in the movie, but now it's become reality. And this is how the field is developing with the fast pace, with the new developments in the with the new scientific development. So the basic question comes in, like, how do they, like the machines, they generate and understand the human language, or there must be something. So it's a whole new branch, like whole new knowledge. I'm sure I cannot cover everything, but I just want to give you the keywords, so that way, when if you want to know further, you can have some idea about where to start from. And I think the large language model, like the LLMs, are they are the deep algorithms, basically very complex set of mathematical complex set. I'm not sure with the life scientist how the math complexity of math goes, but at least I'm not like very much good in the math, so I would leave that in a plain simple language. So that deep learning algorithm basically like summarizes, it finds the pattern, and based on the human inputs, it generates like the data that can be the machine readable format. And when we talk about the data, so like data is basically the brain of AI. So without any data, we cannot develop the AI. So as I said in earlier, like when we do any small observation or any small experiment, that becomes your data. Or from our day-to-day life, if we just start from like looking around, like the weather, transportation, medicine, like our own health, these all things are quantitative, and all these numbers, they are become the data. So today's data becomes tomorrow's AI tool, and that's how it's gonna improve. So what has happened in the past will become like has given us development as of now, and all the tools like ChatGPT or Copilot, they are like using, yeah, they are progressing based on this available data. So it's not like AI is came from somewhere like from the clouds. No, it's like what we have put into the world in this world and through the internet now. Since the information becomes global, it is easy to have access of this data and make interpretation to learn the future. Like weather forecasting is our very known system which uses the past trends and makes the future, yeah, assumption like how the weather gonna be like tomorrow or next, or like the next rainy season or next winter. And since that information is available or based on only local observations, that doesn't give you the real picture. But because of the global data set repositories and the satellite information, now all the global weather forecasting can be unified, and then that gives you more realistic view of how accurate we can predict the weather forecasting. So, and what is important for this is like the data and not just data. And since one more thing we will talk about what can make a bias into this, I will talk about that in the next. So when we think about the machine learning, like the data is like the energy for machine learning. So unless there is any data, we cannot make machines learn about the patterns. So here in this image, so this is the image of eye, like the corneal image, and you can see that these different images have like showing the thickness of your left and right eyes, and also based on the shape and size of the patient's cornea and their thickness over the time, clinicians they generate these numbers, and these numbers becomes like their data to provide for the machine. So one of the interesting projects right now is going in our department is about the diagnosis prediction. Like when we look at this cornea through the through that the lenses, then depending on the corneal thickness, we can predict like what is the health of patient's eye, or like how the disease is progressing, or how the how the treatment is working, whether that corneal thickness is increasing or decreasing. Measuring them every time is time consuming, and like it's not comfortable for the patient for their eyes to hold on for such a long time. So with this data set, the hospital associated with this university has huge amount of data sets, and now they are adding those using that data set for the patient to quickly diagnose their symptoms. So that can give you an extra help for the physicians to save their times and provide the better service in the short amount of time. So overall, how any AI tool develops is like we can see from the sequences, like it all starts from the data points in terms of life sciences. If we consider how the personalized medicines can be developed in the future, like they are already in the pipelines, but if we want to develop in the future, so it all starts from your data from the different different aspect. Like let's say from your RNA sequencing is like the previous generation was like when we had all the NCBI data sets that helped finding the mutations, and that kind of help researchers to know how the mutations are like affecting our life, and then how we can fix those issues by developing the new medicines. But since these things are like more of the individual specific, so over the time we are learning that any disease or any particular mutation affects every individual with their differently. It's not same for all. So one tablet for all is not a like the good idea. So we should always or like based on the scientific evidences, it is now more and more clear that individualized medicine will be needed in the future to make treatment more effective. So we collect data at different time points, like let's say based on your lifestyle, what is your genomic background, what is the phenotype of patient, weight, height, uh, their age, gene expression data set, what the lab resources are showing up, what the imaging, like the diagnosis is showing up. So this all contributes in generating a high dimensional data set that don't just think from one angle, like you have fever, that means you have that disease. No, it's not only that, but it also considers the like the historical data sets. That kind of reminds me, it's a little out of track, but very relevant. So one of the projects here in the UIC was going on was like they basically doing through the neighborhood. So they have basically fixed the satellite positions for the patients in their area. So they measure all the like 24/7, what is the dust particle status, what is the weather, what is like the humidity around their area, like basically the pollution and the state of disease. So how your environment immediately in your periphery is contributing to your disease, and they're developing models based on that, like to show that. And one of the interesting findings where they found that the area where there was like more industrial pollution, people are suffered from more like the inflammatory diseases, and that is when they ask the patients about if they do feel this pollution in their day-to-day life, they say no, it's like very clean, and we don't just realize that what is outside, but it eventually affects your life. That's what is like the major impact. So when we think that uh, if we are suffering from any condition just because of our personal health, so that's not the complete truth. It's also like our environment or other factors do contribute in those situations as well. And then these high dimensional data sets we become used for the learning methods, and that through the high computational methods that data becomes analyzed and finally getting validated with the preclinical studies, and that comes into the market. So there are not just the personal medicine, but the biotechnology can influence multiple key areas. I won't go much in detail, assuming last presentation might have covered it. But I would like to talk about like more of as an example. So I have here 3 examples from 3 different areas, and how they are like these 3 different focuses are improving are like potentially contributing to improve our life. So one of them is like the NVIDIA. So it's not the NVIDIA, they are the chip makers, but also they're helping a lot in the development of AI. But this is NVIDIA, it's like in Veda, so it's like the Ayurveda. So this is like the one of the company here in US, and they do have spread across, and in India also they are working. So basically they are trying to like get the information from the natural plant material. So when we think about the plant material studies, like how difficult or time consuming it is to get the information from plant material, like to extract the components, then characterize them, then identify them, and then see their effect on, let's say, in the bioassay or any health effect. So over the years, people have done like, let's say, like one researcher kind of spends their whole life in developing one molecule from one plant, and that's like the lot of human efforts. So they are simply what they're doing is like collecting all this information available, what resources we have known so far from the plant materials, and learning through the machine learning, they are developing the models that can translate this known information into the better applications. So recently they got like the very venture funding for that, in knowing that how that platform is efficient in identifying the key molecules. So you don't need to do the experiments. Actually, you just need to use the what is available information and put that into the machine context, so that you can reduce your time in referencing the non-essential information, and that basically speeds up the discovery of the druggable molecules. So when we think about the drug discovery and development, so such efforts, like they basically help to accelerate this process. And they also, like through the predictive modeling, they can just like use the efforts, like they reduce our reliance on the trial and error method, and eventually that contributes like the personalized medicine that we discussed earlier. The next I mean, here we can see that what I was talking about, the conventional approach, like it takes long time to develop one active molecule from the plant materials, but with the AI approach, now this big information becomes like at one place, and the machines will bring out this information. You might see something abnormal with these images, and you might be right, like these are like the generated through AI. And as we see, I will give this example of this image later on as well. Like not everything we generate from AI is like the real. So we as a human, we definitely need to check it, like how these things are like making sense or not. So it's just like the grain of salt on top of, no, everything is not like very wonderful. You still need the humans to fix some of the things. And I was happy to see, so this is the last part I joined, and that you already know that the AlphaFold, which has basically revolutionizing the protein chemistry, so which is like very hard to study, resource intensive and expensive. But with this Google DeepMind's collaboration, the AlphaFold becomes like the next thing. And as in the last presentation, we have seen that student can basically just follow the tutorials there. It simply takes 5 minutes, but the amount of the quality of information you get is like, it's worth like the years of efforts, and you basically save a lot of money. I mean, so when we think about the AI, we also need to be like the more realistic and practical. Like when we see that these things are like, okay, very really good, impactful, but we can't just like start and invest right away. We know that we have some economic resources or limitations, but the availability of data is free, and we need to think on where the students can develop their self as a future leader. So just using this free data, you can basically set up your expertise, and that expertise so can give you the future opportunities. So like to say, when you have the data set, you analyze and predict the results for other companies, because then companies won't spend that time again in screening out all the data sets. So without the real investment, you can still contribute in the biotechnological development. So, and it's not just like you need the very like the like kind of in this image, the lab like this. No, that's not like the necessary. I think like the data is like the much is the least I mean, I'm just finding the right words to put it in, but it's like the cheapest information, but it's very valuable. It's like some people call data as a gold, and you're gonna that's because that's implications in the AI. So use that data for your like making some interpretation to find your question. So one of the example I can give you here, like so when one of my experiments, we were studying how the infection can affect the patient, like the health of the mouse animals, and then in those we sent that data for the publication in the Journal of Immunology, and then the reviewers ask us like, okay, this is nice, fantastic work for the in context of animals, but does this happens in the humans? Do you have any evidence that this can also happen in the humans? So I mean, it was true that we have no access to the human data sets, and working on the humans is like whole different set of work with all the like the getting approvals and everything. So it's not like the as simple as it seems. So I mean, we thought that maybe now just because of this aspect, they might not accept the article. But then I realized that there is a data set available on the NCBI. It's like the GeoBus, which has like whole tons of like it's like tons of data sets like through RNA sequencing, single cell RNA sequencing. They are very expensive experiments, but all data set is free. So I thought to, okay, I see these proteins in my data in that mouse model. So are there any data sets that kind of show some information on the like the herpes infection and the humans? And luckily there was one data set from the humans, and that was like the big number, like 25 minimum 25 samples per condition. And that was like the very interesting thing for me, because now I could save all my time in figuring out how to solve this or how to justify my research. When I analyzed the data sets, look for the protein expression in the available data sets, and they were very like aligning with our observations in the animals. And then we provided that new analyzed data sets, and it was just like accepted as it is. So, so without spending any money, basically we provided the evidence, and this is how I was like mentioning that how faculty or student can make use of these data repository data sets in their day-to-day like the research life. So another example I would like to talk about the AI in agriculture. So as you know that, so anybody know about this AgriPilot AI? Is there something I mean, I don't know. It's not like more interactive, and I'm the only speaking, so you might not be uh, able to responding like lively. It helps basically helps in the classroom settings to get more feedback. But the AgriPilot is like the one of the startup from India, from Baramati in Pune. So they and the recently. And they have the collaboration with the Microsoft. So this becomes like the global global startup. So that's how they are using the trends of the crop patterns, crop yield, their diseases, and their and by analyzing all the data sets, so they are generating. So recently in the news, I read about how they are using this startup for studying the sugarcane production to increase the sugarcane production. So what examples I'm giving here is they are not from like only from the developed world. They are just like very close to you. So like right in Baramati, they are using AI to improve the agricultural income from through the agriculture. And I think as a student, we need to be aware about those developments around our area. You can reach out to them, ask for whether you can share some data sets, or they can just as simple as any traineeship to see how it works and how does machine learning process actually work. And obviously, no matter why we are developing this, just to get the benefits like increased yields, like reduce the resources that we invest, and also by more sustainable way when we monitor it more precisely. Obviously, we need to use less chemicals and less operational costs, and that helps the agriculture production. So, I mean, these things they are already allowed, and there could be many other examples, and they're like it's not just like too much to discuss, but when we think about the future, like where it is going to take, and that is like more important for you thinking of how what you are currently learning as a life scientist, uh, how you visualize your future. So I feel like these like the AI driven personalized medicine could be like the more taking shape in the future. Then synthetic biology, like how these vaccines getting developed using the AI. We know that how the coronavirus vaccine was like discovered in a record-breaking time, and also like the epidemic outbreaks, how we are using this information to learn from to predict the future epidemic outbreaks. And also the most important is like the collaboration between AI and biotech researchers. So that is, I think we are at this age where we need to collaborate more with your computer scientist friends and to provide each other a set of information as simple as just trying to develop a mock data. So just if you make the machine learn how to identify the colors, that is like the good enough to start with to get the idea about how these things work. And with that prototype, you can literally develop anything in your day-to-day life. There was an area people developed so many apps to make our life comfortable. And I think with this biotechnological integration, we gonna use more, make life more comfortable too. But as I mentioned earlier, like whatever we see is not like very always good. We also need to think about what are the concerns of these new things. When we do new things, obviously we learn new things, but at the same time we realize that what way we should not use this thing to reduce the non to make it like more comfortable to use in the future. So I think one important aspect is the data quality. So if your data, so what you make the machine learn, if it is not like the realistic or based on the bias and it's not fair, then the machine is like machine is gonna just give you the outcome based on your biased data. So if I mean that you can test yourself that if you just put random numbers and ask machine the question, you're gonna see the random outcome, and that's not really logical. So that logic should come from the humans, and that should be true and honest and based on the ethical concerns. So one of the major ethical concerns people feel about the AI in the facial recognition nowadays. So many places they just scan your face through the retinal scanning, like even the smartphones. So, but that also comes with so when they scan your retina, basically the provider is getting access to your health, and that's also if you are in a like the ethical way, you can make use of that information to help your customer. But on the other side, if that information goes in the wrong hands, that can like compromise your privacy too. So here I would like to talk about a very short story about like the how like the scientists, they discovered one of the sweet drugs. So they were developing that. This was like the initial times. So one of the project was to make the machine learn based on the available information that what makes patients feel good, so they can develop like the good drugs to study. So, and they randomly use all the data from available on the internet through different sources. That was not real scientific data. So the outcome, would you imagine what could be the outcome like that can make person feel good? And it's like, so they their goal was to just bring the wonder drug that can just make you feel good with your disease condition, and the outcome was like the chocolate. So I mean, we all know that chocolate, like everybody loves chocolate, but this is not like the real drug. It's like the temporary gives you temptation and sort of thing, but you cannot treat treat in the sense like the disease person. You cannot treat the disease person just with the chocolate. You definitely need the targeted medicine. So you need to make sure that what the outcome machines are giving are really useful or not, and what you need to provide it better. The other thing in the early era of ChatGPT, when people started getting a lot of interesting information, they were asking what is the reference for this, and there are many evidences, like even I tested that. So it just gave the random references, and if we go back and search for those references, you won't find any publication, but it will be, but the way ChatGPT will give it with like the names, article title, publication title, journal, volume, everything, but it's not really, it's like basically non-real. So you need to like the retest the information what you're getting through AI, and those are like the major emerging issues we need to think. One of the important thought I was thinking, like in terms of money, how it affects to make the machines learn, and basically it's like the electricity is one of the major consumption to make this computer run. So if you think about to run a small, like to do an 1 year study, how much monthly expenses you need. So it costs around $216. So it's almost like kind of close to 20,000 annual just for the electricity uses. So it is our responsibility to use it like carefully, and as like when we talk about the sustainable, then sustainable means it should be accessible to everybody, and we can use it for the longer term. And apart from that, so that the cost, so this is just electricity, just an example, but there are many costs associated with the AI. At some point, people think that the AI is more costlier than the actual production of the current industrial things. But I think as it comes like over the time, the smartphones were early times were like 10 times expensive than their current price. So eventually the price will come down as the users go up. So we hope to have those leverage in the coming future. So, I mean, through this AI and biotechnology aspect, I also would like to share like what knowledge we are generating and how we are integrating these aspects for our scientific understanding. So this is one of the experiments we did in the past to understand what happens in the animals with the virus infection. And so we use the metabolomics. The metabolomics is basically we analyze all the chemical molecules in your given sample with the LCMS analysis, and with the help of statistical analysis, we can basically check whether those molecules are same or different between your different samples. So this PCA plot shows here that that two group of samples were had like the different chemical molecules, and but we didn't know at this point which chemical molecules are making this difference. So to study that, I mean, we also look for what molecules this is referencing to, what is the content and what are the highest molecules in these samples. So we again, how we get this information is based on the available information. So in the past, people have developed this knowledge. They have created this data set about which pathway these molecules are part of, and we basically compare our knowledge with the existing data set to know what our samples are representing, and we narrow it down to one of the pathway, like the molecule which was involved in different pathways. And we took that molecule further in doing the in vitro validation studies, like whether this molecule can really affect the virus infection. So basically, here we just show that, I mean, that was the very first evidence that when this is like the standard molecule, when the infection occurs, this molecule synthesis takes place. But if we inhibit this molecule with the specific inhibitor, then we do not see this molecule. That means your inhibitor is working. And when we add this inhibitor in presence of infection, then also we don't see the production of your molecule of interest. And this is like this imaging basically shows that the color here is like the tagged virus, which represents GFP and RFP. So GFP is for early proteins and RFP for late proteins. And how these in presence of inhibitors, we do see that the expression of these proteins getting decreased. So that was convincing validation of our data that these molecules could be one of the potential candidate drugs for to rescue from the herpes simplex infection. And with this Western blot data, also we can see that the protein levels, actual protein levels of these proteins, they go down when we inhibit them with the inhibitor. And the another interesting thing here was we see that this ICP0 is early protein and the GB is late protein. So this molecule is working on the later part of virus life cycle. So it is more targeting late proteins. And so with this validation studies, I mean, what data we get from the resources, it's not just like we can believe on that, but we need like the better validation to make the stronger conclusion how this data helps for the to make our observations even more interesting and successful. In another set of studies, we were testing that how do the ocular infection can we identify some biomarkers from ocular infection. So usually when eye gets any infection, it secretes the liquid, basically those tears, and that tears, they contain some immune cells. So my focus of my project was like the neutrophils, which is like the major our innate immune cell markers, like the population that responds to any infection happens to eye. So we measured over the time. So these are the times how the infection progresses. So the mock side is like animals were not infected, but the red plots here, bars here, they're representing like how over the time the infection progresses, and the intensity of these neutrophil associated molecules are increasing in the tear samples. And that we tested in like the C57 black pig mice as well as in the diversity outbred mice, which are like more of the represent the diversity in humans. So like as we know that every individual is different, so the diversity outbred mice, technically each mouse has the different genotypic background. So we are using them to represent the better human population and make our observations more like the translational. So in the both cases, we saw that there is some like those immune marker associated secretion is increasing when they are getting infection. And in collaboration with the LV Prasad Eye Institute back in India, we also confirmed that in humans as well, those molecules are highly expressed. So our goal here is to narrow it down, those molecules, through the metabolomics approach. Okay, so first I will talk about, so these are like the human's eye, and this is how herpes keratitis looks on the human eye. So this is very like the uncomfortable disease and can lead to the blindness eventually. So this gives us the confidence that how the mouse studies can be also applied in the humans as well, and if those both observations are making sense, then we can use the ocular fluids as a target to make to develop the more machine learning. So in this, these are basically the like the graphs for human tears and the mouse tears. So they typically shows so the intensity here is like is very similar in the both cases, and their chemical contents are also comparative in the both cases. So that way we can understand that this information could be useful for making like the non-invasive biomarker formation. So now currently we are working on like how this diversity outbred mice, when they receive infection, then we do their untargeted metabolomics. Untargeted metabolomics is just like unsupervised machine learning, where we don't know what we're gonna find. So without keeping any restriction, we just go for whatever is there in our samples. It will detect all the detectable chemical components, and those molecules, through the network analysis, we are narrowing it down to the biomarkers, and those potential biomarkers we are again setting up the experiments to do the validation. And then with the targeted metabolomics, now we know which are the biomarkers we want to target, and those only we going to measure their quantity, and that way we are setting up this model. So with the computational scientist as well, with the Discovery Partner Institute, we are like, and the human samples, with following this data with the different like the following this like, so this is like the tradition, I mean the conventional approach which we can use for the developing any AI model, like from your experiment to the model validation and the prediction, basically in like the currently our studies are ongoing. So but the end goal is like, if I take in the future sample from the eye, just the tear fluid, then I would be able to see, and when I will look for my targeted molecule, like, okay, if I find this molecule in this concentration, that means the patient is in this phase of the disease. So just like the blood test, but the tears are like the non-invasive. You don't need to put any like inject anything. Just a paper strip is good enough to collect your sample. And this is how we are exploring the potential of AI to further bring our research to improve the human health. And yeah, I think to conclude my like the talk, I would just like to give you the sense of like the provocative like the question, like, okay, you basically, how do you imagine your world of AI and biotechnology together to solve your like the greatest challenges you feel right now? And that's what do you see will be the next invention. So I think we as a like the individual, it is more important to be aware and be watchful about what we need, and that's how we can bring out something to the humanity. So, and I think that's what I'm learning through over the years. Like how do processes from thought to the actual product they go. So anything is like we have in hand today was the thought, just the thought in the past. So I think we are in the area right now. It's like kind of the golden age to grow together and further. So please use all the resources that are around. Internet is no longer like the any privilege. You have easy access, and I think if you just learn how you can make use of those resources to better your life, and I think that will be the, I think for any faculty to develop, that will be the greatest thing to see how like how you have developed your expertise for the sustainable development. And with that, I would like to thank you all. I would, yeah, just to finish that, I would like to thank all my like the institutes so far, like NMU, University in Israel, France, and US, as well as like my mentors and friends, those who helped me a lot throughout this journey. Dr. Satish Patil sir, Dr. Lawrence from France, Dr. Zaritsky in Israel, Cedric, Dr. Deepak Shukla is like my current mentor, and Rahul is my like the great friend. He's like helping or like also together we are growing in this field. So that like I think it's always nice to or like feel fortunate to that I came I met these people in my life, and this is how we are like also they are like they helped me to progress so far, and I think uh, like it's also like my responsibility to follow to take that baton, and would like to be helpful in any terms if I can. So please reach out, don't hesitate, and thank you all. Yeah. And thank you again for the organizers for such a wonderful opportunity. I mean, I would like to be there to see all of you in person, but I think when the opportunity comes, we will be to have like more informal discussions. If you have any uh, like questions not related to research or not relevant to the AI, I think AI is maybe you have good enough information and you can also learn a lot from the resources available. But if you have any questions, please let me know. Thank you so much.
So thank you. I request participants, if you have any queries, you can put. Yes, dear participants. I request participants, if you have any queries, you can ask. Are you there, participants?
Maybe it's a weekend evening and that's how, but still, I mean, if good like to have, I think they might be started. Like, I mean, it's good to have him. I mean, I think a session is recorded already, so
Yes.
yeah.
We have recorded the session.
Yeah.
So if there are no queries,
Mm-hmm.
a gratitude for your insightful session on AI for biotechnology research, dear Dr. Chandrasekhar. On behalf of everyone who have attended, I want to extend our heartfelt thanks for your incredible session on artificial intelligence in biotechnology research. Really, your deep knowledge and engaging presentation style really make us a lasting impression. So this lecture inspires us to think us about how we can integrate these cutting edge technologies in our own research. So we are grateful for the time and effort you invested in sharing your expertise. It was a privilege to learn from a lot of things from you and we hope to have the opportunity to host you again in the future. So thank you once again for making this session both educational and inspiring. So thank you.
Absolutely. Thank you. I mean, Sandeep, I'm just calling Sandeep because I think we studied together, so that's not I mean, yeah. So it's really, it was great, and I still remember you were also one of the faculty development program participant when with Dr. Patil sir.
Yes.
At our back together in 2013. So,
Yes.
And he talked very high about you, like how sincerely you are thinking about your students, so in developing like their future. So I know that you are like very serious about your students, and similarly, Dr. Ravi Patil sir as well. So I mean, I think together you are like making this RC Patel College like taking to the newer heights. And once again, thank you so much. And if we can be of any help in any future, we will be happy to just reach out to us. Okay. Thank you so much.
Sure. We'll reach you for any kind of help. Our students.
So can you share your email ID so that we can contact you?
Yeah. So my on the first slide, my email address.
Yes, it is there in title slide.
Yeah. Okay. Just a minute. Yeah, so it's like the CD Patil at uic.edu is my like the working and where I am available anytime. So please reach out to that email address. Thank you so much.
Okay, so thank you.
Okay.
Thank you. So I thank you again, Dr. Chandrasekhar. And thank you all the part.
Now I'm leaving the meeting. Thank you all, all the staff who is making this program successful. Thank you so much. Bye-bye.
Thank you.